of you are Biology 110 students, and so you may have some questions about the independent project, which we will address after the faculty presentations. You have, I hope, each of you a program, and the program contains the research statements of the faculty who will be mentoring projects in Biology 110. So there will not be time for questions of the individual faculty. That will be something for you to do in the weeks that come after this. So without further ado, I'm going to turn over the floor to Karen, and faculty Beth will give us the 3-minute high sign when we're getting close to the end.
Hi everybody, I'm Karen Bagley. I'm broadly interested in how disturbance affects plant and animal communities. I've spent a lot of time studying how fire affects bird and terrestrial salamander communities in western coniferous forests where fire is a key element. Fire is also important in Ohio environments where it's usually used to maintain prairie habitats. It also can have a very profound disturbance of what kinds of plants and animals live in an area, sometimes almost completely changing the ecosystem. An example of disturbance that's going on right now locally is the emerald ash borer, killing a lot of ash trees. We don't yet know what the effect of that disturbance is of the mortality of ash trees on the other organisms in the environment. I'm also interested in climate change in which broad patterns of temperature and precipitation are changed, but it also can have effects on things like fire and insect outbreaks and storms, things we typically think of disturbance events. We can use observed responsive organisms to recent climate fluctuations to project how organisms might change. So say if we project that there's more drought, so we can take information that we know how organisms respond to drought to make predictions. Or we can use models of climate and biological response to make predictions. So you may have seen the work of Professor Kirchhoff's ecology class. This last semester, they did some presentations on it. They used known locations of species from a model called MAGIC, in which you can extrapolate future locations based on their preferences. The assignment brief here, the R code, to do that is that topic introduction. Studying organisms in the environment allows us to understand response in a complex world. But ecological relations in the field can also be complicated. And so we have a lot of ways with study design or statistics for which we can control for variables that are not part of our question. We also have a lot of tools in which we can observe organisms both directly and indirectly. I brought just a couple to show you how we might do these sorts of things. So here I have your classic tools that most people think of, binoculars, terracotta, study birds, but they are also used on things like looking at leaves way up in the canopy or butterflies. This is a live trap in my house. So this is used for small mammals. We put a little bit of bait in the back. There's a little pedal. They step on it. It closes the door. You can use ink and tracks to indirectly measure organisms to find out what's out there. Each kind of animal has a unique track. You use something very simple, low tech like that. This is a board? Yes, it's just a board. You use it for looking at terrestrial salamanders. You can lay these out there. They're like an artificial log. And so then you have known locations which you can look it up. The salamanders go under there. It's nice and moist and dark. They go under there in the day. And you lift it up and you see what you got under there. So those are some things we can do. So I presented a research area that's very broad, but I am here to help you to hone a project into something that's doable and that interests you. Thanks.
Hi everybody, I'm Chris Gillen. I'm an animal physiologist, and I'm going to tell you a little bit about what my research lab does and then how some of that might translate into Bio 110 projects. So I'm a molecular animal physiologist, and I'm interested in how animals respond to challenges to their salt and water balance. My research right now is using mosquitoes, and so I want us all to, for a moment, put ourselves in the place of a female mosquito, so try to develop a little bit of sympathy for the mosquito. So the female mosquito is flying around, her biological clock is ticking, she needs protein to feed, to make eggs, so that she can hatch some cute little squiggly mosquito larvae. And what she's going to do is she's going to take a blood meal. And the blood meal is going to have a lot of protein in it, but it's also going to have a whole lot of salt and water, which for her is more or less useless. And so she's going to need to get rid of that. So mosquitoes, particularly female mosquitoes, are world champion peers. She will expand her body weight by almost twofold, or more than twofold, during that blood meal. And she needs to get rid of all of that. The next thing she's going to want to do is fly before she gets swatted, right? And imagine trying to fly, right? That's hard. Now imagine your body weight was double when you try to fly. That's even harder. So that's going to be a tough deal for her. And in fact, mosquitoes, there's pictures of female mosquitoes actually peeing while they're still taking the blood meal. They start peeing right away. They're world champions. So we study that. What we study is the proteins that are involved in transporting salt across cell membranes. And that's important because it helps them regulate the final salt concentration of their urine. It also is the mechanism by which they produce urine, because they transport salt and the water falls. So we study those proteins that are involved in that process. We use a whole range of techniques, from molecular techniques like RNAi to knock down gene expression and quantitative PCR, to measure gene expression, to biochemistry like Western blotting, to good old-fashioned physiological techniques. We sample mosquito hemolymph, their blood, and we have a machine called a cation chromatographer that will measure the ion concentrations in very small volumes. So, we do a sort of whole range integrated look at that process in mosquitoes. Mosquitoes are small and they're really hard to work with. And learning these techniques or collecting enough anal papillae or malpighian tubules from a mosquito to do something with is going to take a long time. And so they are not particularly amenable to bio-110 projects. But I suggested in the pamphlet that you got that crayfish are a good alternative organism to think about. We can do a lot of the things that we do with mosquitoes, with crayfish. They are remarkably salinity tolerant. So you can put them in almost 75% seawater, and you can put them in fresh water, and they respond. So we can challenge them and study how they're responding both at the physiological and molecular level.
Hi everyone, I'm Karen Hicks. I teach classes in genetics and also in evolutionary developmental biology. My research program looks at the mechanism by which plants use seasonal cues, so day length and temperature, to know what time of year it is, and then regulate their reproduction in response to that, so that they undergo reproduction at a particular time of year. And you guys probably have noticed plants doing this out in the world. Also, lots of animals do this. And we understand quite a lot mechanistically about how it works in flowering plants. So we know a lot about the genes and proteins that are involved in sensing light and temperature cues, and then transferring that information to regulate gene expression in the organism to then lead to production of flowers. And so my work over the past decade or so has turned to thinking about how this might work in other plants in order to understand whether or not this mechanism evolved a long time ago, prior to the divergence of land plants from one another about 500 million years ago, or if it's evolved separately in different plant lineages. And so we've been working on a moss species called Fiscomycella patens. And this is a plant that's really easy to grow in a petri dish and easy to manipulate, so give it different light and temperature sorts of conditions. And the genome has been sequenced, and so we know a lot about the genome of this organism. And so we're interested in asking whether or not the same genes are involved in this moss. And I've been using we've been taking advantage of strains of this moss variety that were collected across Europe that turn out to respond differently to light and temperature cues from one another. And so we've been using those to mine the genome using the whole genome sequences of these strains as well as differential expression to try to come up with a list of candidate genes that we think are involved in reproduction. And so that's really the direction my work is going, and I just recently, yesterday, received funding from the NSF to support this, so I'm really looking to grow my research group. So I'm really excited about having students do projects with me who might want to continue on working on these projects from the IntroBio lab. So one possibility for students in the IntroBio lab would be to take some of these candidate genes and look at differential expression in response to either day length or temperature cues and see whether or not the data that we're collecting from whole genome analysis holds true. Another thing, I'm running out of time, yes. And another thing to do would be to look for new phenotypes in these natural variations. And then the last thing I want to mention is that I'm getting involved in a new collaborative project that also involves Chris Bickford, working with another moss species called sphagnum, which is peat moss. And we're really interested in seeing what conditions sphagnum can be induced to undergo reproduction. So that would be a fun, complete experiment.
I'm Harry Nagaki, I teach cell biology and neurobiology. I'm trained as a central neurobiologist, and I'm always in the business. Over the years, I've done quite a number of different things with students. They include things like the role of a gaseous signaling molecule, nitric oxide, since the nitric oxide in central transduction in flies. I've worked on ants and how their olfactory cues are actually being used to differentiate between different food sources. But right now, the main part of the work has been working on the mid-gut. It's kind of not really particularly neurobiological topic. It's a collaboration that's going on with Dr. Kenyon and Dr. Philpott, looking at the role of size and scaling the size of the gut. It turns out that being large is actually quite a bit different. Small because the surface area volume ratio of the tissues to transport to the surface is actually varies. And we've been looking at expression patterns of certain transport proteins using some chemistry and also Western lab to see whether these actually change. I've also started becoming interested in the enteric nervous system, which is a nervous system associated with the gut. It turns out, at least in mammals, that there are as many neurons associated with the gut as there are on your nose in your stomach. It's also a great unknown for the nervous system because it's really hard to study. Nobody really wants to spend money on it or time on it. And I think there's some interesting things we can actually do in invertebrates to try to see what the roles of these neurotransmitters might be in the gut. And so I actually started some work on it. There are a couple of students starting to work on looking at serotonin and some other neurotransmitters in the recent gut to see whether we can find some physiological changes, whether the expression patterns change over time. And so that's kind of the way I think the work is actually going. As for what the Bio-10 students have done, I've had students working on taste perception in Drosophila using the behavioral assay, which is actually really cool, very cool. You get lots of data very quickly. So you might, for example, ask the question, can a fly differentiate between aspartame, sugar, fructose, glucose, right? And then we can challenge that with different kinds of repellent compounds such as quinine to see where the kind of the tipping points are. That gives you some idea of what the fly actually thinks is actually interesting. How much time? 30 seconds. So I'll tie up now. I'm actually open to quite a number of different kinds of topics, and so come see me if you want. I'm usually a guy in the 108, 109 with dog awareness.
Hi everyone, I'm Drew Kirchhoff. I'm an ecologist, so I study interactions between organisms and their environment, broadly defined, much like Professor Bagney was talking about. I'm also a biogeographer, so I study the distribution of organisms across the face of the earth and how biodiversity varies over space and time. And so in my lab, we have a number of different projects going on. Some are related to the ecology class that I teach every fall, looking at the dynamics of carbon in some local forests, and I have some projects related to that I'll talk about in just a minute. But the main research that goes on in my lab is in the area, in sort of an emerging area of biology that's known as eco-informatics. And so most of you have probably heard somewhere of bioinformatics, so the use of large data sets to understand molecular systems and genetic systems. I work on eco-informatics, which is the use of large data sets to understand ecological systems, and particularly to understand biodiversity. So the main project that I'm involved in, and much like Professor Hicks, I was recently funded by the National Science Foundation to do this, so I'm looking for students to work on this in my lab, is a collaborative project with researchers at the University of Arizona, Wesleyan University, and then an international network of researchers called, known as the Botanical Information and Ecology Network, to utilize large sets of data to understand the distribution of plant life over the face of the earth. In particular, we're focused on North and South America. We have distributional, functional trait, and phylogenetic information, so the evolutionary relationships, for somewhere on the order of 85 to 90,000 plant species in North and South America. And we're interested in everything from their evolutionary history to their projected responses to climate change. So if you're interested in large data-driven research projects, come and talk to me. There's lots of sub-projects that can be carved off for 110 to do that work. A lot of that work is also driven by trying to understand what causes variation in biodiversity. Here, very near Kenyon, I have a forest plot that's about 2 football fields in size, that has about 20 species of trees in it. The same size plot in Costa Rica, where I've done some research, has about 200 species in it, so about 10-fold more. We're trying to understand why there are those gradients in species diversity. It very, very closely co-varies with climate, but what exactly the cause is not clear. I also have some ideas for field-based projects. Some of these are related to the forest carbon budget study that I mentioned earlier. I have equipment that's known as infrared gas analyzer that can measure changes in CO2 concentration to understand movement of carbon from the soil layer in the forest into the atmosphere. That's how forests interact with the atmosphere, and they're a very important player in the carbon budget and in climate change. So I have opportunities to work on projects related to that, in particularly thinking about how during the spring season, when you're going to be doing this project, how do ecosystems wake up? When do they start generating carbon relative to when they start taking up carbon through plant growth and photosynthesis? So that's an opportunity. The other opportunity that I'm interested in looking at is in the evolved characteristics of plants, in particular aspects of their xylem anatomy, so the anatomy of their vascular system, how they move water and nutrients from the soil up to the top of the tree. There are some very interesting relationships to examine there, and also thinking about characteristics of the leaves. So at the same time during the spring, as the ecosystems are waking up, that's also when spring ephemeral wildflowers are emerging from the leaf litter layer, and trees are starting to leaf out. Some trees leaf out quite early. Are the characteristics of these trees different from the trees that leaf out later? So one of the projects that I have in mind for 110, if you're interested in field work, is to examine some of these sort of early starters in the spring season to see if their leaf characteristics, their thermal properties and the physical properties of the leaf that are related to photosynthesis are different from the leaves that come out later in the season. So just to wrap up, I have a number of different sorts of projects, both data-driven and field-based, that are available in my lab. I'm looking to take on probably 2 to 3 groups. So if you're interested in this kind of thing, since I'm not an instructor in the course, the availability of my lab is going to be a little bit limited. So do come and talk to me soon so we can start thinking about papers that you might read in preparation for the projects and so on.
All right, everybody, hello. So my name is Erin Linstead. I'm actually visiting here this semester from Ohio State, where I teach as well. And so I don't have an active lab here currently. So if you guys choose to do a project with me, we'll be doing some work on some of the stuff that I was interested in, did my PhD on. So I want you guys to kind of picture in your mind your favorite organism or an organism you like, and don't everyone picture a panda because there are other things besides just pandas out there in the world. And so I want you guys to imagine that that organism is faced with an enormous number of challenges in its life. It has to find resources, so mates and food and territories, while simultaneously avoiding predation. And so this might sound like seem like, that's what's the big deal. Well, you can't really devote all of your phenotype to gaining resources because then that comes at the cost of being able to avoid predation. And you can't really devote all of your phenotype to avoiding predation because then you miss out on all of those other important aspects of life. And so animals their phenotype reflects this interesting balancing act between trying to gather resources while also simultaneously avoiding predation. And because predation is such a ubiquitous and certainly strong selection pressure, we see that animals have evolved a variety of really, really cool responses to predation. So we can think about something like a turtle whose morphology seems to be almost entirely devoted to avoiding predation. That's a pretty extreme example. But we can think about things not just morphologically, but also behaviorally. So something like a Pomeranian antelope that's one of the fastest land animals that we have. Thompson's gazelles are some of the fastest, and they are currently hunted by, of course, the fastest animal. And so we can think about those suite of behaviors that are directly involved in that predatory interaction. So a cheetah chasing after a gazelle, there are some behaviors involved in that. But like I mentioned at the very beginning of this talk, there are a suite of behaviors that are not directly involved in avoiding predation, but that are really, really important to an animal's survival. And so those are the kinds of things that I'm interested in looking at, is how does that risk of predation affect other kinds of behaviors? So things like foraging. So, an animal has to forage. Is their foraging behavior at all influenced by a risk of predation? So we can alter the riskiness of the environment. We can make them think that an environment is particularly dangerous or not particularly dangerous, and see how they forage. Does that affect how they change how they forage? Do we have to give them a bigger foraging reward in order for them to take greater risks, in order for them to, say, leave the cover of some kind of nice shelter? I'm also interested in looking at things. One of the main questions I asked for my PhD was looking at learning. So animals have to learn about their environment all the time. Are they more likely to copy the decisions of other animals if the risk of the environment is high? So if it's a really risky environment, do you just say, I'm going to stay here under the cover of shelter and watch what everybody else does and go ahead and just assume that that's the right decision and copy what they do? Turns out that might be in fact the case. It looks like there's more mounting evidence that that's true. Also things like sexual conflict. So we know that males and females often diverge in their interests when it comes to mating, when it comes to rearing offspring. Does the risk of an environment affect that? Do they become more aligned in does sexual conflict reduce? Does sexual conflict increase when we change the riskiness of the environment? And then one of the things that I was really, really interested in, the thing that I'm actually the most interested in, is looking at behavioral flexibility. So we can think about all of us, or you can think about your dog or your cat. We know that our animals don't always behave the same way day after day. They exhibit a repertoire of behaviors. But what's interesting is we can think about how does the risk of the environment change the repertoire of behaviors that they might exhibit. So it might be that if you live in a particularly risky environment, or if you're faced with predation, selection favors you to behave the same way over and over and over again, in some successful way. You might also predict that if you're in a risky environment, maybe selection favors you to behave differently all the time in order to confuse predators. This is the idea of behavioral flexibility and how it responds to predation risk is something that I am really interested in. And certainly, if you are working under me, then those are some questions that you could ask. So what I worked with historically is mosquito fish. They are little freshwater fish that are really easy to keep in the lab. We can manipulate their environment. We can trick them really easily into thinking that they live in a safe or a non-safe environment by just altering how many plants there are in there. If there's lots of plants, they think it's a safe. Exactly. If there's lots of plants in there, they think they're really safe. If there's no plants in there, they think that they are now suddenly exposed to the predator, which isn't actually there. But that's okay. They think that. So it's really easy for us to manipulate that risk and then look at those behavioral differences. Does their behavior change? In what ways does it change? Oh, excellent. Okay, thanks.
So hello, I'm Professor McMahon, and although I don't have a research program per se, I have a long history of dealing with plants and their poisons. So if you think about a plant, and you're not a plant biologist, you may think of them as very sessile creatures that really just sit there and don't do very much. But I'm here to tell you that plants are dynamic organisms, and they respond in very unique ways to the environment around them. Because they're rooted to the ground, plants can't just run away from their problems the way we do, and they can't go to counseling. So what they have to do is they have to evolve some way of responding to the stresses of the environment in a way that will allow them to continue to live. Now, one of the ways that plants can respond to the environment is through chemical production of a vast array of different kinds of chemical compounds, an arsenal, if you will. So again, these plants are not just sessile, they're getting a little offensive, and they're going to fight you and other things in the environment with chemicals. So the stresses that plants can go through are both the biotic kind, say an herbivore who's munching on a leaf, or they can be abiotic, such as excessive heat or excessive light. So plants have evolved these different kinds of chemical responses depending on the kind of stressor that is being applied to the plant. So one such chemical response is something called cyanogenic glycosides. These are a class of compounds which have a glucose molecule attached to a moiety that has evolved from an amino acid, and the amino acid has this special nitrile group. What happens in the plant is that these cyanogenic glycosides get stored away someplace safe, like a vacuole or a cell wall. And what happens is that these compounds just stay there happily, not hurting anybody, until somebody hurts the plant. As soon as the tissue gets damaged in some way, say, again, an herbivore bites into a leaf, what you have is the cyanogenic glycoside is suddenly put in the same environment as the enzyme which can break it down. And you've created a cyanide bomb inside the plant. So imagine, if you will, the cyanogenic glycoside comes into contact with an enzyme and releases a whole bunch of hydrogen cyanide. Is that good or bad? Cyanide is bad, right? It can mess up your molecular respiratory machinery. So this is a way that plants can deter herbivores by this threat of chemical compounds. There are lots of other projects we can do in my lab besides cyanide, but what I suggest that you do is you read my bio in the manual and read some of the review articles, and you can think about different kinds of stresses, different kinds of plants, and different kinds of chemical defenses. Thank you.
Hi everyone, I'm Sarah Peterson and I'm recovering from a cold, so hopefully you can hear me. I teach in both the biology department, biology 109, 110 is here, as well as the neuroscience department. And that's because I do have an interest in neuroscience, though my interest in neuroscience is very fundamental. I'm largely interested in how genes drive your development. And so I apply that largely to the nervous system. So nobody's used the board, so I'm going to. So if you can imagine, you've got a neuron right up here in your spinal cord. It's a motor neuron, and it needs to grow from the perspective of the neuron all the way out here to your feet or to your hands. And it's got to innervate those muscles in your hands in order to make your fingers wiggle. And this process is called an axon, and it's going to have to traverse an incredibly large distance for that relative to the neuron in order to be able to innervate that muscle. And not only does it have to interact with all of the environment along the way, it's got to know where it needs to make a target, and it's got to persist in making that target, called the synapse, that connection between the neuron and the muscle. It's got to continue to make that synapse as the body grows and changes. So obviously you have experienced this, but you're also experiencing it right now in your brain as you're making, as you're learning things and making new memories. So that's also a form of synaptic rearrangement. So both the development as well as these changes during the life of an animal, are controlled by your genes and your proteins. And so I'm interested in understanding how your genes influence development and the changes that you experience later in development in your nervous system. So obviously it's very hard to do very controlled experiments in humans on the genetics of the nervous system. And so in my very new research lab, this is my first year at Kenyon, we are using the zebrafish model organism, which develops very rapidly, and as opposed to your approximately 100 billion neurons that you have, it has in the order of millions. But in order to study development, you have to have embryos, and my zebrafish are not yet happy enough to provide embryos to you in a 110 project, which I would like to be successful. And so for the purposes of Biology 110, I want to switch gears and study an organism that I used back in my PhD work, which is Caenorhabditis elegans, better known as C. elegans. And so C. elegans are these microscopic roundworms. They're about as thick as your eyelash and about 1 millimeter long. And they have precisely 302 neurons that make very precise connections. And that's important because if you want to understand how genes influence development, you want to be able to manipulate the genes in a way that show stereotyped changes in their development. So what you would do in a Biology 110 project is use C. elegans in order to manipulate gene function in some way. There's a number of ways you can do that. There are available mutants that show nervous system phenotypes, and you can also knock it down with something called RNA interference or RNAi. You simply feed it to the worm and it stops having that gene protein product produced. And so using C. elegans are pretty simple animals. You can do a lot of fluorescence microscopy work. Unfortunately, I don't have all of that equipment quite set up yet. And so all you really need to do is see if their basic functions that they use the nervous system for are effective. So basically C. elegans crawl and reproduce. If part of their neuromuscular junctions that I said at the very beginning are messed up, they no longer crawl and they no longer reproduce. So you can very easily assay whether or not a gene is controlling the function of the nervous system. So if you're interested, please come talk to me. Thank you.
Good morning, I'm Wade Powell, and I guess I'm one of 6 or so of the life sciences faculty who work in the area of molecular and cellular biology. My training is in the regulation of transcription, that is the production of messenger RNA. And I have, for the past 20 years or so, studied a particular transcription factor called the aryl hydrocarbon receptor. So it's a receptor, it binds other chemicals and specifically environmental contaminants. And it's the binding of contaminants to this receptor and the subsequent changes in gene expression that cause toxicity in animals. So I've especially been interested in amphibians and the evolution of the structure and function of this protein in vertebrate animals generally. From a technical standpoint, I'm able to help Bio 110 students work on projects in a wide range of organisms using a common set of techniques to measure gene expression, RT-PCR, Western blotting, things like that. And I've helped students use various invertebrates, use plants. I'm especially interested in tadpoles and amphibian metamorphosis. So if any of these organisms or technical approaches appeals to you, I'm probably a good person to talk to.
Hi, I'm Dr. Schultz. I am a physiological ecologist, which means that I study how an organism is impacted by its environment and specifically looking at its physiology. Within the field of physiological ecology, I study ecoimmunology, which means that I look at the immune system. So the immune system can vary based on abiotic and biotic factors. Specifically, an organism only has so much energy that it can allocate to any one process. And so, for example, if it's investing a lot in reproduction, it can't invest as much in its immune system. The environment, so things like weather, disease potential, so how much disease is in the environment, nutrient availability or food resources can have an impact on the immune system. age, sex, the condition of the animal. There's a whole species can also have an impact on the immune system. So immune system is something that's really critical. We all need it to survive, but because it is costly, it can vary. And so all those factors that I just talked about can influence how and when it varies. So I look at birds, or so I work on birds specifically. So what I envision for these 110 projects is going out to the BPEC and actually catching birds there. So we would catch a lot of different species using mist nets. From those birds, we can take a blood sample. And from the blood, you actually can get a lot of information. So we can look at the immune response. We can look at, for example, different white blood cells, both the quantity and the type, because that can tell us a little bit about what's going on. Additionally, we can, for example, mix the bird's blood with a known quantity of bacteria, plate it, and see how well the bird's blood gets rid of that bacteria, which is a measure, we can use that as a measure of immunocompetence. Additionally, we can measure things like hormones, testosterone, which is the main reproductive hormone in males. We can look at how that would co-vary with the immune response, which can, testosterone often dampens the immune response. So if you're interested in birds and interested in going out to the BPEC and catching and taking blood samples, I would be the person to work with there. So there are many things we can look at for projects. We can look at things like age, how that impacts the immune response, how species, different species, how they would vary. And we can also look at things like within an individual, how do the different immune responses, how do they co-vary within an individual. So those are just a few ideas, but there's obviously lots of possibilities. Okay, thank you.
So hello, I'm Dr. S, and I represent the bacterial lab community at Kenyon, the students at Kenyon who are studying bacterial genetics and genomes. And so much of the work we do is on a model system, Escherichia coli, but for Bio 110 projects, we can actually look at bacteria that are involved in any kind of host organism. We now know that all organisms, including multicellular organisms, have a microbiome, which includes all the associated microbes with that organism. And we have the tools to investigate questions. For instance, what bacteria are associated with your dog, with different parts of the skin of your dog? Do different parts of the dog have different microbial communities? So in a Bio 110 project, we can investigate that. Or what about the prevalence of drug-resistant bacteria? So which of your friends harbor drug-resistant bacteria? You can get handprint from all your friends, and we'll take a look at the species and drug resistance of those bacteria. So we can look at those kind of questions in our 110 project. We can also look at different environments if you want to do environmental sampling of microbes. If you're interested in a more long-term project, you can get involved in the work we do funded by the National Science Foundation on the question of stress response in bacteria and drug resistance. We have a project looking at the use of aspirin to modulate the microbiome to lose drug resistance. This is a very exciting published project, and we look at the genetics involved in that. That's more long-term. So either way, there are lots of things. Basically any kind of idea or organism you want to work with, there's probably something we can look at.
I am Chris Bickford. I'm a physiologist here in the department. And if you all are interested in that dog project, I have a dog. So for my lab, we are interested in plants. And it's just, we've got an idea today. Plants are complicated. Plants are sessile, but they are complicated organisms. Specifically, I'm speaking about leaves and the feature of the leaves. We saw my seminar earlier in the semester. You know that I'm interested in trichomes. Trichomes are the hairs that you find on plant leaves and other organs, but I'm interested in leaf trichomes. They're intimately involved in defense, so they keep insects and things like that from munching on leaves. But I'm actually interested in the other aspect, how they influence the physiology of leaves. Right? These are physical structures, so they are doing something to influence how, say, radiation is impacted on a leaf. They may influence its carbon relations, they may influence its water relations, and the latter two factors are what I'm interested in.
in terms of 110 projects, the things that I've been thinking about that are relevant to the timescale you all are operating on, really kind of think about this. How does the environment influence trichome density in a plant? We don't know much about that. We know a lot about the genetics and the molecular biology of trichome development, but we don't really know what in the environment triggers trichomes to develop. So that's something that's relatively simple, but still relatively unexplored. Another thing that can be explored is the relationship between trichomes, their density, and how they influence water relations on a leaf. So you might not think about it, but if you think about the difference between a lotus leaf and a rose petal, which are two things that you might or might not have experienced, their interaction with a droplet of water is quite different. Right? There are physical-chemical differences in those two leaf surfaces that actually influence whether the leaf droplet will stay on the leaf or whether it will be shed off of the leaf. Okay? That may seem superficial and not important, but actually in functional ecology of plants, that's actually quite important. And we can talk more, if you're interested in that, about why that's important. This is a relatively simple thing to test in a number of plant systems in the time frame that you have. I'm also interested in if folks are interested in kind of more broad or less specific stress responses to plants. So if you're interested in kind of growth and how plants might respond to a particular stressor that you're interested in, talk to me and we can maybe think about a way that you could approach that. Okay? So I have 2 slots open. So the thing about plants is, unlike E. coli, they don't grow quite as quickly. So we can't just start one day and 2 days later be ready to roll. There's a little bit of lead time needed, so we want to have a conversation sooner rather than later in order to get those projects rolling. Okay? So please, you're welcome to come talk. Thanks.
Okay, so that concludes our faculty presentations. What I want to do is talk to the students for a second so you have some ideas of what the next step is for you. The preliminary project ideas are going to be due 2 weeks from now. So that means that starting right now, you should really start seriously considering why you're taking biology and what kind of research questions are intriguing to you. So you have some guided reading in each of the faculty bios that you receive. Use those as a starting point. You also, as Professor Bickford said, you want to have early conversations with the faculty because many of them have limited spots in which to host research. So especially the faculty who are not teaching Biology 110. So have early conversations. You will also be working with somebody in your lab as a lab partner. We don't want to have 110 different Biology 110 projects going on. We probably will have about 40 to 50 projects happening. So the faculty in 110 will start trying to match students who have similar research interests. So as you move forward, you can work together on the ideas, the formulation, the research, and then eventually the project itself. So in the coming weeks, identify faculty members, multiple ones perhaps, who might be able to work on projects with you. Once you have some project ideas, approach those faculty and have conversations with them. In class, we will have conversations about the project and start to see if we can't put some of you guys together as research partners. The project preliminary proposals are in, and the description of what's required. This is all in the syllabus. So make sure you read the syllabus if you have questions about the independent project and the various steps that you're going to be going through. Part of the syllabus contains important dates for the independent project. Pay attention to those dates as well. So are there any questions that you have right now that perhaps we can answer? This is a slightly different format than what we have done in Biology 110 in the years prior to this. So many of us are still trying to figure out how all of this is going to work as well. Are there any questions from any of you at this point? Not yet? Okay. Then thank you very much for attending and have a great day.